Hello, thank you for joining. It's great for me to be here. Today, I want to share with you a story around structure, dynamics, and function of endocytic vesicles with the STELLARIS Confocal platform. The vesicles I'm going to focus are here. You see them in this movie. They are these fast-traveling structures that you see in red, and they are the central players in what we call the endocytic pathway. We know that these vesicles are not just swimming inside the cells, but they shuttle material by interacting with multiple organelles and cellular components, and they do it in a very dynamic way. This dynamic aspect, of course, is a common theme of life, and using different light fluorescence imaging approaches, we can find answers from a structure going to molecular mechanisms to reach a mechanistic understanding and a functional understanding of a given process, going from molecules to organelles to organisms. And I hope to show you in this presentation how different tools and modalities integrated in our STELLARIS Confocal platform can help you in this quest across scales. So to give you an outline of my presentation, first I will focus on the vesicles experiment and show you how we can image them at the right speed and quality. Then I will show you how we can get functional information from this pathway using a set of special tools. After that, I will expand into how to build a full picture from even more angles, exploiting our STELLARIS platform capabilities, and a brief summary at the end. Let's start on how to get the required speed and quality that we need to study the trafficking in these vesicles I've showed you. Going back to the time lapse that you saw at the beginning, we have here 4 fluorescence labels in live cells. We see the actin filaments in magenta, we see microtubules in yellow. We can also observe very clearly the mitochondria in cyan, and the cell membrane and internalized vesicles are displayed, as I mentioned, in red. We are capturing this information in high quality and detail, enabled by key components of our confocal. These key components are our next generation white light lasers, a full reworked beam path, our spectral detection concept, and the new Power ID detector family. Our STELLARIS platform was designed with the goal of maximizing photon collection in the mildest conditions to enable gentle live cell imaging. And now, in the next slides, I want to go more in the technology details that enable these benefits. So I will start with the detection capabilities behind the STELLARIS detectors. The Power ID family is built on 2 main architectures. We have the Power ID-S that are based on silicon multipixel photon counter technology, and here the fluorescence photons are spread onto a detection area comprising an array of independent detectors of units. Each one is a silicon avalanche photodiode, which are all wired in parallel and read out over a common anode. Around this MPPC technology, we have engineered a unique concept to get superb image quality. We worked on a 3-stage calling scheme to suppress dark noise and on the optomechanical design to ensure maximum photon collection. The Power ID-S can operate in analog and in photon counting mode, and it is our all-round high-performing detector. It delivers excellent sensitivity in the blue-green spectral range that is key for some of the most widespread live cell fluorescent protein tags, and it also provides a broad overall detection range that extends out into the NIR range, so the near infrared, up to 850 nanometer. I will show you later in the presentation that this extended range is great in combination with the excitation provided by our white light laser technology. Now let's move on to the other members of the family of detectors on Stellaris. We have the power high DX and the power high DR. These two detectors are based on hybrid detector technology. So we have here photocathode front plate, that is gallium arsenide phosphide for high DX, and extended red gallium arsenide phosphide for high DR. And they are coupled to an avalanche diode. The readout for these detectors is fully digital and photon counting based with virtually no dark noise. All the three types of power high D detectors, as I mentioned, enable photon counting, that is the mode of measuring signal coming from a sample when one wants to be quantitative. And leveraging from the photon counting properties of our detectors and the electronics of Stellaris, we have introduced power counting. Power counting is a proprietary concept for identifying and counting photons that otherwise we would miss from our experiments. So what happens in the photon counting regime, we can detect single photons, and we will characterize this single photon detection by a given pulse width. So in principle, we can also detect two photon events separated in time. So we will have the pulse width defining these photon detections, and we will have also a second parameter to characterize, that is the pulse pair resolution, that gives us the temporal spacing in the arrival of the photons. So in conventional counting, what it can happen is that when we are detecting photons, when we have a second photon arrival that is below the pulse pair resolution, then this second photon will be missed from the counting because it will be confused and integrated with the first photon that was detected. With power counting, what we can do is correctly account for the second photon by using the pulse width information. If we now look at images of different cellular structures that were recorded with or without power counting, what we get is an increased signal, and this happened for all the three types of detectors that we have for the high DS, for the high DX, and for the high DR. And we also get an extended dynamic range while staying and being quantitative. And that means brighter, crisper images and more detail. And talking about quantitation, we can translate the benefits of power counting by looking at recorded signal as a function of the excitation in intensity. So what we see here, the power counting, is the red solid curve. We see an enhanced dynamic range compared to conventional counting, where the linear response in conventional counting is really at this low level. We have published a Nature Methods application note with the principles and applications around the power high D family, and this application note is open access and you can find it very easily, and we can share the link with you to go into the details and learn more. I mentioned earlier that one of the ingredients to get great quality images was our new generation of white light lasers. So this is the second key innovation embedded in Stellaris that rounds up the capabilities of the detectors with optimal excitation, maximizing the signal. Combined with the AOBS, acousto-optic beam splitter technology, we have a system of ultimate flexibility and less complexity because we have a single laser that can do the work of many. We can apply up to 8 single excitation lines simultaneously, and we have a huge and extended range of wavelengths to choose from. In the case of the white light laser present on the Stellaris 5 and the Stellaris STED system, we have excitation capabilities from 488 to 685 nanometer with an optional NIR extension up to 790 nanometers. And in the case of the Stellaris 8, we can go even from 440 nanometer excitation up to 790 nanometer. And this means that you can perfectly match the excitation according to the fluorophore or the fluorophores that you want to use. And a key point here, and a very interesting one, is that the white light laser range for all members of the Stellaris portfolio can offer excitations, as I mentioned, up to the NIR range, which offers extra benefits combined with the NIR detection capabilities of the detectors, of the power high-D detectors. And this allows, for example, to very easily realize multicolor experiments like here, where I can show you 5 colors simultaneously with 3 of them in the NIR range. So this was recorded with Stellaris 5, and Stellaris 5 can now deliver at least 3 more colors in the NIR range, as an example. So with this, I have covered the image quality aspects that we need to address, signal quality and temporal resolution that we need to study vesicles in the context of the cell. So now I want to go into the second part of my talk and focus on getting functional information on top of the speed. As we know, fast processes happen at many different levels in the cellular environment, and to study them, we must be, of course, at the right speed, but that is not enough. We also want to learn something, and that comes hand in hand with understanding what's going on, and that implies understanding and getting to grasp the function of the molecules. And that is why speed needs a special extra, and to show you this, I will go back to the traveling vesicles, although this time I will start with this time lapse that shows fast transport in action. One thing I have not mentioned yet is which labeling we are using here, and in this case we have labeled the cells with an NIR membrane fluorescence probe, and we captured the fast vesicle movements with a speed of 1 volume every 1.8 seconds. These kind of experiments are typical when one would perform studies on endocytic pathway, which is super relevant to many different processes like cell dynamics, signaling, growth, and also host pathogen interactions. That is a very hot topic nowadays because many animal viruses enter host cells via endocytosis. During endocytosis, we have endosome maturation. Now, a hallmark of endosome maturation is the pH changes that take place as the maturation is happening, going from pH 7 to 4 from the cytosol, early, late endosomes, and finally reaching lysosomes. And this is where function comes to play. If we can sense those changes in pH, we can understand what's going on. So we need to find a way not only to visualize the vesicles, but also to get a readout of the pH changes. And now you can see that the limitation is not only and not on the speed, but on the sensing, because if we just restrict our measurement to the photon counts that we detect, that is the intensity, we don't have a huge revelation of what is happening about the functional state of the vesicles, because the pH changes are not reflected here. So here I have a zoom-in region of the movie, and we can track the vesicles, of course, which we can watch them come closer, even fuse, but that's it. And here is where we have a very special set of tools that kick in, this extra ingredient I was talking about. And we have developed these tools because we know that one aspect of fluorescence is the intensity, but there is an extra dimension, which is the lifetime dimension, and we want to access that on our confocal. And we want to do that because with lifetime we can sense environmental changes, and that can give us access to functional information to characterize, in this case, endosome maturation. So here we go. The first special tool that I want to show you is tau contrast, where we use the average arrival time of the photons, and that is lifetime-based information, to reveal what the vesicles are doing. So now we have the speed and we have the pH sensing information because the NIR probe that we are using behaves differently at different pH values. And that is the environmental change that we are exploiting and mapping as a color palette, together with the intensity that we already got from the confocal experiment. And this happens simultaneously and from the same measurement that we are acquiring. And we can do more. Using a second special tool called tau separation, we can interpret the lifetime-derived information in more detail, and we can generate different channels according to the different response that the probe senses from the different pH values. With some more work, this can be translated into different maturation steps. And this is how mapping of the lifetime dimension looks like with this extra layer of information. We can, here, as we see here, we can overlay the channels, and now we see the vesicles in different colors that correspond to different functional states. And now if we go to the zooming region, we can now see the vesicles in different states traveling and fusing. We already had the speed to follow the process, and now we have added the insight into the functional state thanks to these special tools. And as a bonus, as I mentioned, because we used an NIR label, this means that we have all the rest of the spectral window, the standard, let's say, spectral window, free to choose other labels that are relevant to the application that one is interested in investigating, and leveraging, once again, from the flexibility of the excitation and detection of STELLARIS. These tools that I explained, tau contrast and tau separation, are part of our new tau sense technology in our STELLARIS confocals. Again, you can find more information on this technology, on tau sense, on a Nature Methods application note, and here is the address. We will share it with you so you can get more details. So now with this example and with these results coming with functional insight, we can do even more. And for that, we will use AVIA. So I want to highlight in the next slides this extra powerful possibility that comes with the options available in the AVIA software package combined with these results from the tau sense experiments. So with AVIA, we are capable of now tracking the individual vesicles not only at the intensity-based level of information, but indeed using the tau-separated channels in which we have staged the different maturation levels. Here we have this information and we can analyze it downstream to look for mechanistic hints of the maturation process. So what you can see here now is that the different channels and the different steps in maturation, so how these tracks look like. And now for the analysis of the tau separation separated channels, we have included a large number tracks per channel, so you see them here, and we got different metrics related to each of these tracks. So with these metrics, we can now put numbers to the behavior of the vesicles in different functional states, and this could, for example, serve as criteria to compare how the process is affected upon external perturbations, upon stress, and integrating information coming, for example, from experiments where we address up and down regulation of signaling proteins. So here what I have selected is the mean track velocity, and what you can see here is the distribution of this parameter, and what we get here is a hint that there may be two populations in play, and now this is worth further investigation down the road with the rest of the analysis and integrating complementary information. Finally, I want to bring a final topic into this presentation and change gears and enter the last part of my talk, in which I want to discuss and show you how to build the full picture from even more angles. And this brings me back to one of the first slides, because we have developed our Confocal with the idea in mind that you can find the answers to your questions with complementary approaches in a single system. So our Confocal is the tool for addressing your science and not a bottleneck. And I selected as complementary modality our latest approach to STED, with the aim of characterizing further the endocytic vesicles. And this is inspired by a unique combination of STED and lifetime in our system, and that is what we call tau-STED. And before we go into the application example of the vesicles, I want to explain you why we developed tau-STED. And to do that, I selected this example on this different sample that shows very nicely the essence behind our development. What we have here are nuclear pores immunostained with Alexafluor 555, using an antibody that targets the pore basket. We expect to get point-like structures, but their sizes are way smaller than the diffraction limit, so we cannot really resolve them. And if we think of the live cell imaging experiments I have shown so far, what we aim to do, one would like to approach STED in a very gentle light dose condition approach. So we apply that idea here in this sample, and for that I just tweaked the Alexafluor 555 with a tiny bit of STED light, let's say 2%. In conventional STED, this is what we get as a result. We can get a bit of improvement to count the pore units, but this is really low quality and we gain little structural detail. And this is how the same image at the same gentle light dose looks when we apply and when we use tau-STED. We protect the sample, we reach great STED quality, and we increase the resolution greatly. This means we can apply tau-STED to resolve the structural details now on our vesicles, going from confocal to tau-STED, as you can see here, where we have vesicles, in this case labeled with CF594, and we also have a second label, sertubulin, that we use to label the microtubules. We can also apply this approach dynamically, as you can see here, and in the context of STED resolved microtubules. And we can see here dynamically the traveling and the dynamics of the vesicles, and we have the possibility of tracking the vesicles and characterize the motion as they travel along the cytoskeleton. And we have done that, and you can see here in this gallery how we can track the movement and assign and pinpoint the position of the super-resolved vesicles over time. So I mentioned that tau state is possible thanks to the unique state and lifetime capabilities of our Stellaris system, and the question now is why this is so. Why is lifetime-based information so powerful? And the answer lies in the heart of the fluorescence emission process. So let's take a closer look, and for that I have this simplified Jablonski diagram in which we can see the excitation and the emission process when molecules are excited from the ground state to an excited state, and then they will get rid of this energy that we have given them through the emission of a photon when we have fluorescence. And this emission is characterized by a number of photons emitted, and we can measure an intensity, and this is what we see with the emission of fluorescence. But also another characteristic is the time that it takes for the molecules to go back to the ground state, and this is what we assign and what we measure as fluorescence lifetime. And fluorescence lifetime is very powerful if we have access to this information, because what happens is that fluorescence lifetime gives feedback for competing processes. So whenever we have an alternative road to get rid of this excess energy, the fluorescence lifetime will change. This is what happens when we have, for example, an environmental change, as we have seen in the example of the endosome maturation. But in this case, the competing process is the STED process. The fluorescence lifetime will change because of the STED process taking place. So now let's think about what we see with STED and lifetime imaging. When we do an STED experiment, what we do is we have an excitation provided by, in our case, in our system, by a pulsed white light laser excitation, and then we overimpose the STED donut. And by applying this strategy, what we generate is an effective STED PSF that in the intensity domain will look like a sub-diffraction effective PSF. And when we have access to lifetime-based information, the way that we will see this STED PSF is as a tau-coded STED PSF, in which we can now map these differences in lifetime-based information as a gradient in the values associated with the lifetime that we are measuring. And so this means that we can map the behavior of and the STED effect along the scanning process in our system, in our scanning system, on a pixel by pixel basis. So having access to this lifetime information reveals relevant information for a STED experiment. And the way to access this lifetime information has been realized in different ways. In a seminal approach, it gave rise to the gated STED approach, and later on we also have early inspiration by pioneering work that was done to access this lifetime level of information. And we have here the pioneering work of Vici Domini, and also work done by other groups, in which we see that using the phaser flame approach, we can map this different behavior in lifetime. And in the case of what happens with STED, if we have this mapping, so this visualization of the lifetime based information, what we have is for the confocal information, we will have a cluster or a cloud of information in our phasor plot. And what we will have is, for the STED process, we will have a STED, what we call a STED trajectory that you see here as this elongated cluster, in which the longer lifetimes that correspond to the center of the donut, when we have the maximum resolution provided by the STED approach, are towards these longer lifetimes, and then we go as the STED effect is applied and up to the crest of the donut, we have a decrease in the lifetime of the fluorophores. And also, of course, what we have is uncorrelated information that is present in the form of background noise and uncorrelated signal. And Stellaris is equipped to have access to lifetime-based information in the form of thousands, in the form of this technology and a set of tools to address different aspects, as we have seen, in the form of tau contrast, tau separation, to study the endosome maturation and get the functional insights on this process, but also the same technology that serves for this purpose and gives access to lifetime-based data also enables tau-STED. So how does it work? What we do for a tau-STED experiment is we scan the sample and we collect fast lifetime-derived information at each scanning point of the STED acquisition, and we send this information to the FPGA. At the FPGA level, what we will do is identify the STED trajectory and generate an FPGA tau-STED phasor. And we do this on the fly at the FPGA. So actually what we will see, so we will not see the phasor because this will be automated and done at the system. And a key difference with other type of approaches is that we don't need to get the confocal information to get the tau-STED image and integrate the lifetime information for our STED experiment. And as a third step, we will obtain the tau-STED image, as I mentioned, on the fly and then in an automated way. And with the tau-STED output, we will be able to get an image in which we have suppressed background based on lifetime readout, and this is thanks to a tool called background. And also we will be able to increase the image quality as a signal to noise and resolution, and this is with the second tool called tau strength. So our approach to STED with lifetime-based information works on the fly and automated and unbiased. The benefits of tau-STED spread to all kinds of different samples, and I have some applications to share with you now. This is an example of a non-cleared mouse kidney section immunostate for two proteins, synaptopodin and nephrin. And this is a particularly challenging sample because the structures are really, really compact. And what we can see here, when we go from confocal to tau-STED in 2D and in 3D, is that with tau-STED we can increase the resolution and the image quality in 2D and in 3D with multicolor applications, and as is in this case the localization of the two different proteins within the sample. Of course, we can push the limits of resolution, and we can do it with tau-STED in 2D and in 3D. So in this case we have here in the left a technical sample, gata beads of a nominal diameter of 23 nanometer, and we can see that we can really push the resolution while keeping an excellent signal in 2D for the Gatavit resolution and assessment of the diameter. And in 3D, I have here as an example the nuclear pores again stained with Alexa Fluor 555, and you can see in 3D how the resolution and the signal quality increases. So with Tausted, we can reach resolutions better than 30 nanometer in 2D and 100 nanometer in 3D, of course, depending on the sample and the fluorophore that we are using. And finally, as another example of STET now applied for gentle imaging, what we see here is a time lapse of over 200 frames that is performed at really low laser powers for the STET 592, so 10%, and we have imaging at 1 frame per second on live act and neon green that has been transfected into cells. So we can track and follow the cytoskeleton dynamics with STET resolution over time in gentle conditions. So with Tausted, we protect the sample for this kind of applications as well. And as a summary, with Tausted, we can remove the background using a physical readout that is the lifetime information on our sample. We can push the resolution beyond the limits of conventional STET, and we can apply gentle STET conditions for extended imaging. So with this technology, we hope to open the door to new insights into structure and function. Further information with technical details on our technology are again in a Nature Methods application note that you can download as it is open access. And with this, I've reached the end of my presentation, and before going to questions, I want to close with a visual summary. We have seen how the lifetime-based tools provided by Tausend's report on the function of endocytic vesicles, and these tools are also applicable to different systems, for example here to the dynamics and to identify different structures in nematostella. We have seen how the next generation wide-eye laser with AOBS, the spectral detection and a fully optimized beam path and new detectors contribute to excellent quality and to keep up with the speed of fast dynamic processes while keeping gentle imaging conditions. I have not gone into this topic because of lack of time, but we also have embedded tools to increase the insight and get even more detail and extract more information with lightning, with resonance scanner capabilities, and with new tools powered by AVIA. We have a user interface for intuitive and easy to set up experiments, just focusing on the sample and on the fluorophores that are present in the sample. We have the LASIX navigator to explore the sample and get into the details after exploring it and getting an overview of the sample in general. And also we have different modalities. I went into details showing you how Tausted can help for live imaging in gentle conditions with excellent resolution and signal to noise. And we also have multiphoton capabilities with DIVE. We have a full quantitative film approach powered by Falcon, and we have also light sheet through DLS, and we also have CRS options in our system. Finally, I want to thank you for your attention. I want to thank my colleagues that have greatly contributed to this presentation. I'm happy now to answer your questions and have a great discussion. Thanks again.